Hello lovers, in this video Lauren, my expert examiner, is going to be taking you through topic 3 of your AQA GCSE biology, so infection and response. Anything she wants you to pay attention to in exam, she is going to point out to you. As well as this, you can get all the flashcards, all the questions to really help you revise our predictive papers for this year and Lauren's walkthroughs of the predictive papers.
Communicable diseases can spread from one organism to another because they are caused by pathogens that can be transferred. Pathogens are microorganisms that cause disease. One example of a pathogen is a virus. These are small, acellular, which means they are not made of cells. They live inside cells and replicate, causing damage to the cell when they burst out. They can reproduce rapidly inside humans. Another example of a pathogen is bacteria. These are prokaryotic cells that can reproduce rapidly inside humans. Bacterial cells release toxins which cause damage to cells inside the body, which is what gives you the symptoms and makes you feel sick. Another example of a pathogen is fungi. These can grow on the surface of leaves or skin, and they feed off living tissue. And lastly, protists. These are single-celled and often parasitic organisms that live inside and replicate inside host organisms, which cause damage. Communicable diseases can be spread through the air, through direct contact, or through vectors. An example of transmission by air is known as droplet infection. This is where we inhale pathogens in particles from coughs and sneezes. There are two forms of direct contact, either by consuming contaminated food or water that contains the pathogen, or by touching or having contact with infected tissue, for example, blood. An example of contaminated food is Salmonella, which is a bacteria that causes food poisoning. And an example of direct contact through infected tissue is HIV, which can be transmitted through bodily fluids, including blood. An example of vector transmission is malaria. So this is caused by a protist pathogen which is transmitted by a mosquito vector when it bites humans. There are three examples of viral diseases that we have to know. The first one is measles. The symptoms of measles are kind of a red rash, so spots on the skin, and a high temperature or fever. It's spread through droplet infection because it also causes a cough, and it can cause blindness and even death, especially in children. The only way to prevent the spread of viruses like measles is through vaccinating young children. The next example is HIV. This is a virus that infects and kills white blood cells. It weakens the immune system and eventually leads to AIDS, which is where a person has a weakened immune system and will get secondary infections such as pneumonia or colds or bacterial infections, which can then kill them because their immune system is too weak to be able to fight them off. This is transmitted through sexual contact or bodily fluid contact. So one way of preventing the spread is by using condoms as a barrier method of contraception. The tobacco mosaic virus is an example of a plant viral disease. It infects plants and it destroys their chloroplasts in the leaves. This creates a mosaic pattern of discoloration on the leaves and reduces the plant growth due to lack of photosynthesis. You can prevent the spread of the tobacco mosaic virus by burning the whole plant and disinfecting any tools or hands that have touched the plant. And it can be spread by insect vectors such as aphids, so killing those can also help prevent the spread. There are two bacterial disease examples we need to know. The first is gonorrhea, which is a sexually transmitted infection, so we can prevent the spread with condoms, similar to HIV. The symptoms caused are discharge from the genitals and pain when urinating, and it can also cause a fever. Salmonella is the second example. This causes food poisoning that we have already mentioned, as it's spread from eating contaminated food, for example, raw chicken. The symptoms are sickness, fever, and diarrhea. We can prevent the spread by washing our hands before and after touching raw meat, and also cooking food to high enough temperatures to kill the bacteria. In the UK, chickens are also vaccinated against salmonella to try and reduce the spread and make sure it's not present in eggs as well. In both of these examples, the symptoms are caused by toxins released from the bacteria. Treatment for any bacterial infection involves antibiotics. These are drugs that kill bacteria in the body or stop them from replicating. Not all antibiotics will be able to kill all bacteria. We now have an example that there are many resistant strains of gonorrhea, in the UK specifically, which cannot be treated with most antibiotics. There is one fungal disease example that we need to know that infects plants, and that is rose black spot. The symptoms are suggestive of the name, so black or purple spots on the leaves, and the leaves can also turn yellow and fall off. Plants often suffer from lack of growth because of lack of photosynthesis due to these black spots and yellow areas, meaning there's less chlorophyll. It is spread through fungal spores that travel by wind or water, mostly in rain droplets. They can also be spread on gardeners' tools and hands as they work between plants. One way of preventing the spread is to move an infected plant away from the others until it is treated and safe to move back close to them. We can spray plants infected with rose black spot with fungicide in order to kill the pathogen. We can also remove infected leaves and burn them to kill the pathogen and its spores. These two will both help to prevent the spread of rose black spot to other plants. There is only one example of Protist disease that we have to know, and that is malaria. Malaria is caused by a Protist parasite that can live in both mosquitoes and humans, so some of its life cycle is spent in both organisms, and it can cause cycles of fever and it can be fatal. Ways to prevent the spread of malaria include preventing yourself from being bitten. This could mean using mosquito or insect repellent or mosquito nets. Or we can prevent mosquitoes from breeding to reduce their numbers by covering standing water, which is where they lay their eggs and their larvae develop. Human defense systems. Humans have some non-specific defenses that try to prevent pathogens from entering the body. The nose contains hairs and mucus to try and trap pathogens. The trachea and bronchi in the airways contain cilia on the cells to move mucus, which contains trapped pathogens, to the nose and mouth to be removed from the body, or to be swallowed. The stomach contains hydrochloric acid to kill bacteria in food or swallowed mucus. And the skin acts as a physical barrier to prevent the entry of pathogens everywhere else. Specific defences work inside the body in case pathogens do enter, and they're normally carried out by white blood cells. Phagocytes detect and engulf pathogens and digest them with enzymes. Lymphocytes are triggered to divide by mitosis when they detect a foreign antigen. They will form plasma cells, which release antibodies that are specific to the antigen on that pathogen, and they can also produce antitoxins that can bind and neutralize toxins released from bacteria. Vaccines contain a dead or weakened form of a pathogen, so they will not infect the person, but the antigens on the pathogen will bind and trigger white blood cells to produce antibodies that can bind to the specific antigen on the pathogen. So you can see I've got my pathogen, my antigen, which is kind of the spiky protein on the outside, and my purple antibodies, which can bind to that antigen. The white blood cells will form memory B cells that will remember the antigen on the pathogen and will produce more antibodies faster next time they are reinfected in order to prevent illness. This makes people immune to that pathogen. So the first time you are infected with pathogen or an antigen from a vaccine, we go through the primary immune response to make antibodies for the first time. So not many antibodies are made, and it happens slowly. The second time, you get infected with the same antigen. So if you inhale it naturally, for example, through droplet infection, we have what's called the secondary immune response. So because you have memory cells, they produce the antibodies faster and there's more of them. And so we get that bigger peak in the graph that we can see here for the secondary immune response. This prevents you from getting sick and gets rid of the pathogen from the body before it has a chance to make you ill and before you can pass it on to other people. Vaccinating large numbers of a population can prevent the spread of a disease because making people immune means they cannot be infected with the pathogen and so they therefore cannot pass it on to other people. This is known as herd immunity. Examples of painkillers include ibuprofen, paracetamol, morphine, and codeine. Painkillers are taken to treat symptoms of diseases such as pain, inflammation, and to lower temperatures caused by fever. Painkillers do not treat the cause of the disease because they do not affect pathogens. Examples of antibiotics include penicillin, amoxicillin, and tetracycline. Antibiotics are drugs that kill bacteria. They treat the cause of the disease by killing the pathogen. Antibiotics cannot and do not work on viruses because these replicate inside cells and bacteria live outside of cells. So antibiotics cannot get into or work inside cells. Antibiotic resistance. Some bacteria in a population have the ability to survive treatment with an antibiotic. These will survive and reproduce, passing on the genes that allow them to survive to their offspring. Over time, we get an increase in frequency of the resistant bacteria in a population until we have a whole population of bacteria that cannot be killed by that antibiotic. This is an example of natural selection and therefore evolution of bacteria. We need to be able to suggest ways to reduce antibiotic resistance. We can reduce prescriptions of antibiotics for viral infections, so prevent doctors from giving out antibiotics unnecessarily. Making sure that patients take the full course of antibiotics every time, so they take all of the antibiotics they're given, even if they start to feel better. And reduce the use of antibiotics in farm animals. Originally, drugs were extracted from plants and microorganisms. There are 3 examples we have to know. One is aspirin, which is used as a painkiller, and this was extracted from willow tree bark. Digitalis, this is a heart medication that's extracted from the foxglove plant. And penicillin, which is an antibiotic, which comes from the penicillium mold, which was discovered by Alexander Fleming. New synthetic drugs made in the pharmaceutical industry can still have plant extracts as their base, but they must be tested for 3 things: toxicity, efficacy, and dosage. Toxicity is referring to whether or not it has serious side effects or is toxic to cells. Efficacy is whether or not the drug works to act in the way that we want it to. And dosage is about how much drug needs to be taken for it to work whilst not having too many side effects. We need to know the 3 stages of drug trials, what happens in each, and what they are testing for. The first stage is carried out in the lab and it is known as preclinical testing, where we test the drugs on cells, tissues, and animals. At this stage, drugs are being tested for toxicity and efficacy. In the second stage, we move on to clinical trials, which involve humans. In clinical phase 1, we use healthy volunteers and low doses are used to check for side effects. In clinical trial phase 2, we would use patients who are sick with whatever the drug is supposed to help with, and then we are testing for efficacy and dosage. Drugs cannot proceed through these drug trial stages unless they pass the checkpoints. So only if a drug is not toxic and found to work in the preclinical stages will it pass onto the clinical trials with humans, and only if the drug passes the clinical phase 1, where it's shown to not have serious side effects in humans, would it be given to patients, because it is unfair to give or risk patients' health or making them worse by giving them a drug that could cause serious side effects or that does not work. Clinical trials should be at least blind clinical trials. This is where we use a placebo or a fake drug as a comparison to show that the real drug is actually having the effect. In blind clinical trials, only the patients do not know if they are getting the real drug or the placebo. In double-blind clinical trials, neither the patients or the doctors know who is getting the placebo. This is the best standard of clinical trial. Double-blind clinical trials are important because they reduce the potential bias from the doctors. It reduces the psychological effects, for example, the placebo effect, where patients may just report feeling better because they think they are getting the real drug. And it can show that the real drug is having an effect when compared to the placebo. So we know for sure it is the drug that is working and actually having a measurable effect on the illness. Monoclonal antibodies are produced from a single cloned cell. They are all identical and will bind to only one specific antigen or protein. First, a mouse is injected with an antigen. Then, mouse lymphocytes in the spleen are stimulated to produce antibodies that will bind to this antigen. Human tumour cells, which are rapidly dividing, are combined with the lymphocytes to form hybridoma cells. The hybridoma cells divide rapidly and produce the antibodies. Millions of the monoclonal antibodies can then be collected and purified. Monoclonal antibodies have many uses. They can be used in diagnostic tests, such as pregnancy testing, where they will bind to the HCG hormone. They can be used to test blood for the presence of hormones, antigens on pathogens, or other proteins. They can be used to identify molecules in cells or tissues by binding to a fluorescent marker. So you bind the fluorescent marker to the antibody, and the antibody will be specific for a certain molecule you're looking for to see if it's present in a cell or a tissue, and when it binds to it, it will glow. They can be used to treat cancer by delivering radioactive therapy drugs directly to cancer cells by binding only to cancer cell antigens. This prevents the radioactive chemicals damaging the rest of the body. These drugs work by preventing cancer cells from dividing or killing them. You do not need to know in detail how all of these examples work, but any example question or method that involves monoclonal antibodies will involve them binding to antigens or proteins. They will be specific, so they will only be able to bind to that one antigen or protein that they are made for. You can attach identifying molecules or drugs or chemicals to the other end of the antibody. So that means that when it binds to what you're looking for, you can have, for example, a glowing chemical or a radioactive drug or some other kind of chemical like a blue bead, which is what's used in pregnancy testing, attached to the antibody as well. We need to be able to evaluate the use of monoclonal antibodies. The pros are that they can be produced quickly, they can bind to almost any substance, they can help to quickly diagnose disease, and they can help to target cancer treatments directly to cancer cells and reduce harm to the rest of the body. The cons are that they are very expensive to produce, they create more side effects than we first thought due to interactions in the body, they're not able to be as widely used as we first hoped, and they are time consuming to produce in the first instance. There are also ethical objections to the use of monoclonal antibodies, specifically the use of animals needed to produce them because it includes an operation to remove their spleen or cells from the spleen. Also, there's issues with clinical trials where monoclonal antibodies have been tested on humans and have caused serious side effects. Plant diseases can be communicable and they can be caused by bacteria, fungi, viruses, and also they could be infected with insects like aphids, which feed on the phloem. These insects can also be vectors of other diseases as well. Plants can also be affected by deficiency diseases. This is where a lack of a certain nutrients can affect growth. There are two examples we need to know: nitrates, which are needed to make proteins for growth, and magnesium, which is needed to make chlorophyll. Without magnesium, plants can suffer from chlorosis, otherwise known as yellowing of the leaves. Whether the disease is caused by a pathogen or a deficiency, lack of light absorption can reduce photosynthesis, which results in poor growth. No sugars are produced, so there is little energy, and other molecules like proteins cannot be made. So in both cases, plants suffer from poor growth. Plant symptoms that can be identified can include stunted or malformed growth of leaves and stems, spots on the leaves, which we saw with rose black spot, areas of decay or rotting, growths or discoloration to the leaves or stems. This is what we also saw with the tobacco mosaic virus. Here's an example of a necrotic lesion in the bark of a tree where some of the tissue is rotting away. There are some ways that we can identify plant diseases, either by looking at pictures or using a gardening manual or website to compare pictures of your plant with plants on the internet or in these manuals, taking tissue samples to a laboratory to be tested, or using home test kits that contain monoclonal antibodies, which could identify certain pathogen antigens. Plants have certain types of defences that can help to protect them. Physical defences include cellulose cell walls, a tough waxy cuticle on their leaves, and bark, which is layers of dead cells that are around stems that can fall off. Chemical defences include antibacterial chemicals, which obviously can kill bacteria, and poisons to deter herbivores from eating them. Mechanical defences include thorns or hairs on their leaves to deter herbivores, leaves that can move when they are touched, and mimicry to try and trick animals. So mimicry is when an organism looks like another organism.
Ouch! This is why in some videos I explain scratches.